Fenbendazole acts as a moderate microtubule destabilizing agent and causes cancer cell death by modulating multiple cellular pathways. Nilambra Dogra, Ashok Kumar, and Tapas Mukhopadhyay. Additional article information. Associated data. Supplementary materials. Abstract. Drugs that are already clinically approved or experimentally tested for conditions other than cancer, but are found to possess previously unrecognized cytotoxicity towards malignant cells, may serve as fitting anti-cancer candidates. Methyl N-6-phenylsulfonyl 1H benzimidazole 2YL carbamate fenbendazole FC, a benzimidazole compound, is a safe and inexpensive anthelmintic drug possessing an efficient antiproliferative activity. In our earlier work, we reported a potent growth inhibitory activity of FC caused partially by impairment of proteasomal function. Here, we show that FC demonstrates moderate affinity for mammalian tubulin and exerts cytotoxicity to human cancer cells at micromolar concentrations. Simultaneously, it caused mitochondrial translocation of p53 and effectively inhibited glucose uptake, expression of GLUT transporters as well as hexokinase, HK2, a key glycolytic enzyme that most cancer cells thrive on. It blocked the growth of human xenografts in new/new mice model when mice were fed with the drug orally. The results, in conjunction with our earlier data, suggest that FC is a new microtubule interfering agent that displays antineoplastic activity and may be evaluated as a potential therapeutic agent because of its effect on multiple cellular pathways leading to effective elimination of cancer cells. Introduction. The importance of microtubules in cell division, motility, intracellular trafficking and their role in modulating cellular shape according to the environment has made them one of the most successful targets of anti-cancer therapy. Agents that perturb the microtubule dynamics have been widely used in cancer treatment 104. Considering the relative success of mitotic agents in the treatment of cancer, microtubules may be termed as one of the best cancer targets identified till now. 5. Microtubule targeting agents can be broadly classified into two major classes. The first class consists of microtubule destabilizing agents, which inhibit microtubule polymerization. This class of antimitotic drugs includes several compounds such as the vinca alkaloids, vinblastine, vincristine, vinorelbine, vindazine, vinflunine, estromastine, colchicine and combratastatins that are being used clinically or are under clinical investigation for cancer treatment. The second class is comprised of microtubule stabilizing agents. These agents include paclitaxel, docetaxel, epothalones, and discodermalide 6. The consequence of disrupting tubulin and microtubule dynamics with both these classes of drugs in dividing cells is metaphase arrest and induction of apoptosis. Fenbendazole, methyl N-6-phenylsulfonyl 1H benzimidazole 2YL carbamate, is a broad-spectrum benzimidazole anthelminthic approved for use in numerous animal species. 7. Repurposing of veterinary drugs showing promising results for human use can result in considerable time and cost reduction required to develop new drugs. Fenbendazole is known to have a high safety margin and most species tolerate it very well. It has very low degree of toxicity and high degree of safety in experimental animals 8-12. In this study, we show that fenbendazole, FC, exhibits a moderate microtubule depolymerizing activity towards human cancer cells, but possesses a potent anti-tumor effect as evident from in vitro and in vivo experiments. Our results indicate that FC exerts its anti-tumor effect through the disruption of microtubule dynamics, p53 activation, and the modulation of genes involved in multiple cellular pathways. FC treatment also resulted in reduced glucose uptake in cancer cells due to downregulation of blood transporters and key glycolytic enzymes. Since the process of tumorigenesis involves a number of genes and proteins altering various cell signaling pathways, single-target drugs show limited efficacy and may lead to drug resistance. 13-15. Agents having multiple cellular targets, therefore, are expected to have improved efficacy besides the ability to circumvent the likelihood of developing resistance. Overall, the present work demonstrates a pleiotropic effect of FC on cancer cells leading to cell death. Thus, FC may have a potential therapeutic application. Results: FC destabilizes tubulin network in human NSCLC cells. Benzimidazole carbamates have been reported to inhibit tubulin polymerization and disrupt microtubule function in parasite cells 16-17. Results from in vitro studies using enriched extracts of helminthic and mammalian tubulin have suggested that tubulin is the primary molecular target of the benzimidazoles 18. Therefore, to examine the effect of Fc on mammalian microtubule network organization, human non-small cell lung carcinoma, NSCLC, 549 cells were treated with 1-Fc for 24H and processed for immunofluorescence using alpha-tubulin antibody. Colchicine was used as a positive control. Results showed that Fc treatment caused a partial alteration of the microtubule network, figure 1a. The microtubule cage around the nucleus appeared to have lost its intactness when compared with the control mock-treated cells. However, this modification in the organization was not as marked as in case of colchicine treatment, which showed complete depolymerization of microtubules into tubulin subunits. This data suggests that Fc causes distorted microtubule framework of the cells. Figure 1. Fc treatment alters tubulin network of human cancer cells. 549 cells were treated with 1-Fc or 50-ml colchicine for 24H. Following treatment, the cells were processed for immunofluorescence using anti-alpha-tubulin primary. The effect of Fc on tubulin polymerization was further evaluated by an in vitro assay. Purified bovine tubulin was incubated with Fc, and tubulin polymerization was recorded over time. The results showed mild inhibition of tubulin polymerization by Fc in vitro, which was not as pronounced as in case of colchicine treatment. Figure 1b. Next, the effect of Fc on tubulin polymerization was compared with that of other microtubule destabilizing agents like nocatazole and colchicine. Polymerized and soluble fractions were prepared after 24 hours drug treatment, and Western blot was performed using alpha-tubulin and beta-actin antibodies, figure 1c. Tubulin bands of polymerized and soluble fractions were quantified after normalization with their respective beta-actin bands, which served as an internal control, figure 1d. There was a modest decrease in polymeric tubulin in Fc-treated cells as compared to control untreated cells, whereas polymerized form of tubulin was nearly absent in colchicine-treated cells. The result confirms the relatively mild tubulin depolymerizing activity of Fc as compared to other known microtubule disrupting agents like nocatazole and colchicine. A major limiting factor of taxanes and vinca alkaloids is their dose-limiting toxicity and susceptibility to multidrug drug resistance, MDR, occurring commonly due to the high expression of P-glycoprotein, PGP, MDR1, 19-20. Overexpression of beta-tubulin isoforms and mutations are also known to confer resistance to taxanes 21. Unlike taxanes and vinca alkaloids, agents targeting the colchicine binding site are advantageous in that they show minimal multidrug resistance in addition to their ability to overcome the effect of beta-tubulin isoforms overexpression 22-24. However, the major drawback of colchicine and its derivatives is their acute toxicity to humans 22-25. Therefore, a microtubule inhibitor that binds to the colchicine binding site but has low toxicity can be highly efficacious 26-27. The result of a fluorescence-based competitive colchicine binding assay suggests that Fc may bind to the tubulin at the colchicine binding site, Fig. S1. Tubulin acetylation has been associated with the stability of microtubules. Therefore, to examine the acetylation status of tubulin following treatment, human NSCLC cells were treated with different microtubule targeting agents for 24H, and the cell extracts were subjected to Western blot analysis using AC-alpha-tubulin specific antibody, 6-11B1. As shown in figure 1e, while nocatazole, colchicine and vincristine resulted in a marked reduction of acetylated tubulin, Fc did not alter the amount of acetylated tubulin as compared with control mock-treated cells. This result further confirmed the relatively mild effect of Fc on mammalian tubulin as compared with other known microtubule depolymerizing agents. Fc is not a PGP substrate or inhibitor. Development of drug resistance is a major concern in cancer treatment. Multidrug resistance, MDR, caused by the overexpression of the MDR1 gene that encodes P-glycoprotein, PGP, is a critical mechanism of drug resistance which results in a cross-resistance to multiple classes of drugs 28-29. A large number of commonly used chemotherapy drugs like taxanes and vinca alkaloids are PGP substrates 30. However, efforts to inhibit PGP have not shown encouraging results due to unavoidable side effects 31-32. Therefore, discovery and development of novel anti-proliferative compounds that are not substrates of PGP is an effective approach to overcome drug resistance. To test whether Fc is a substrate or inhibitor of PGP, we investigated cancer cell growth inhibition by Fc in the presence of PGP inhibitor verapamil. The results showed that inhibition of PGP by verapamil did not enhance the inhibitory effect of Fc on cancer cell proliferation, figure 2c. The fluorescent dirhodamine 123, Rho123, is a well-known reference PGP substrate frequently used to determine the PGP inhibitory potential of drugs 33. No significant difference in Rho123 accumulation was observed between control untreated and Fc-treated cells, implying the absence of any interaction of Fc with PGP. Fig. 2a-b, in the presence of verapamil, the treated and untreated cells showed comparable levels of Rho123 accumulation, affirming that Fc is not a substrate or inhibitor of PGP. Figure 2. PGP inhibition has no effect on Fc-mediated cell death. A549 cells were left untreated or treated with 10-Fc or 10 verapamil for 6H, or H123 was then added and fluorescence images were acquired after washings with PBS as described under. Fc treatment results in early G2/M block accompanied by cell death. Since inhibition of tubulin polymerization blocks cell cycle progression and may induce mitotic catastrophe, the effect of fenbendazole on the cell cycle progression was examined. A549 cells were synchronized by serum starvation for 48H and treated with 1-Fc for different time intervals. During cell division, cyclin B1 binds to cyclin-dependent kinase 1, CDK1, allowing the transition from G2 to mitosis. Thereafter, progression from metaphase to anaphase requires ubiquitination and proteasome-mediated degradation of cyclin B1 induced by anaphase-promoting complex, APC, 34. Our results show an early elevation of cyclin B1/CDK1 levels in Fc-treated cells, 8H as compared to 16H in case of control untreated cells, figure 3a. In addition, histone pH3, SER10, an indicator of mitotic progression, was used to evaluate whether Fc-induced cell cycle arrests specifically in the mitosis phase, as seen in figure 3b. P-histone H3, SER10, was found to be upregulated at 12 and 24H post-Fc treatment in A549 cells. This data confirms that Fc causes cell cycle arrest in the mitotic phase in human NSCLC cells. Cells in different phases of cell cycle and those undergoing apoptosis were quantified by flow cytometry. The number of apoptotic cells increased in a time-dependent manner with simultaneous decrease in cyclin B1 levels, and 30% cells had undergone apoptosis after 32 hours of Fc treatment, Fig. 4a. The cyclin B1 levels went down further at 40 and 48H of treatment, corresponding to a further increase in the percentage of apoptotic cells. Thus, a decrease in cyclin B1 levels, with a concomitant increase in apoptotic cell death suggests that A549 cells apparently underwent mitotic exit followed by cell death in response to Fc treatment. Figure 3. Fc causes early elevation of cyclin B1 levels and induces mitotic arrest. At I and A2, 549 cells were synchronized by serum starvation for 48H and then left untreated or treated with 1-Fc or 50N colchicine for the indicated. Figure 4. Mitotic arrest followed by cell death in response to Fc in human NSCLC cells. A and B, cells were treated after synchronization as before and FACS analysis was done to detect the fraction of populations in different phases of cell cycle, as well as apoptotic. While a significant apoptotic population, sub-G1 phase, was observed after 32 hours Fc treatment, in contrast, colchicine-treated cells underwent polyploidy at the same time point, figure 4b. Fc enforced cells to accumulate in mitosis, leading to a dramatic increase in apoptosis without evidence of progression to a G1-like status. Figure 4c shows apoptotic cells following 24H Fc treatment. Overall, this data suggests that Fc causes cell cycle arrest and mitotic cell death, consistent with its effects as a microtubule inhibitor. In our earlier work, we reported the absence of any effect of broad-spectrum caspase inhibitor ZVADFMK on Fc-induced cell death 35. The present data suggesting mitotic cell death caused by Fc offers a possible explanation for this result, since mitotic cell death can be caspase independent and is known to remain unaffected by the caspase inhibitors ZVADFMK36-37. Tumor cell lines with wild-type p53 show enhanced sensitivity to Fc-induced apoptosis. Treatment of H460 and A549 human NSCLC cell lines with 1-MFC significantly reduced cell growth as determined by MTTSA, Figure 5a. Tumor cell lines having wild-type WT-p53 appeared to be highly sensitive to Fc action as compared to p53 mutant or null cells, Figure 5b, indicating enhanced apoptosis-inducing activity of Fc in the presence of WT-p53. Consistent with this data, p53 null H1299 cells exposed to Fc following transient transfection with WT-p53 construct showed enhanced apoptotic cell death, Figure 5c. Remarkably, Fc showed less toxicity towards primary epithelial cells cultured from rat lung tissue as compared to a lung cancer cell line, Figure 5d. Wild-type p53 target genes were induced upon Fc treatment, Figure 6a, suggesting that Fc-induced WT-p53 protein is transcriptionally active. However, Fc could also induce p21 independent of p53, Fig 6bc, which may be via its effect on the proteasomal pathway as reported earlier 35. DOS-dependent Fc treatment led to increased nuclear accumulation of WT-p53 in these cells, which correlated well with enhanced apoptotic activity, Fig. 6d. Tubulin acetylation has been reported to enhance stress-mediated translocation of p53 to the nucleus where it is transcriptionally active 38-39. Hence, enhanced p53 nuclear accumulation is concurrent with earlier data showing steady levels of acetylated tubulin in Fc-treated cells. Altogether, evidence supports a proactive role of transcriptionally active p53 in augmenting cell death following mild microtubule disruption by Fc. Figure 5. Fc-mediated inhibition of cancer cells in vitro is affected by p53 status. Human H460 or A549 cells were plated onto 96 well tissue culture plates. Cells were left untreated or treated with different doses of Fc for 48H. Cell viability was? Figure 6. Apoptosis and p53 induction in human NSCLC cell lines following Fc treatment. Western blot was performed for p53 target genes after 24 hours of Fc treatment in H460 cells. H1299 cells were transiently transfected with p53 expression construct. Fc induces p53 mitochondrial translocation. We earlier reported decreased mitochondrial membrane potential, release of cytochrome C and PRP cleavage in cells undergoing apoptosis in response to Fc treatment. It was shown that Fc treatment resulted in accumulation of p53 protein mainly due to inhibition of proteasomal degradation, and an accumulation of the ubiquitinated form of p53 was also observed 35. We, therefore, looked at the mitochondrial levels of p53 when NSCLC cells were exposed to the drug. For this p53 null H1299 cells were transfected with GFP-p53 construct and its subcellular distribution was examined following treatment with different microtubule targeting agents for 24H, Figure 7a. Western blot for p53 showed an increased level of p53 protein in the mitochondrial fraction following Fc treatment, Fig 7bc. Additionally, the p53 translocated to mitochondria in response to Fc treatment appeared to be monobiquitinated, indicating that the increased pool of monobiquitinated p53 in Fc-treated cells is translocated to mitochondria, resulting in the mitochondrial cell death pathway in these cells. 40, Fig. S3. Correspondingly, mitochondrial membrane depolarization was also observed in Fc-treated cells using JC1 voltage-sensitive dye, Figure 7d. Figure 7. Fc treatment results in increased p53 translocation to mitochondria. H1299 cells were transiently transfected with GFP-p53 expression construct and treated as before. They were then stained with red mitotracker dye, molecular probes, and fluorescent. Inhibition of glucose uptake by Fc sensitizes cancer cells to undergo apoptosis. In studies with parasites, the anthelmintic effects of the benzimidazoles have been related to inhibition of glucose uptake with resultant alterations in glucose metabolism 41. We tested the effect of Fz on glucose uptake in human cancer cells. H460 and A549 cells were treated with 1mFc for 4H and uptake of fluorescent glucose analog 2-NBDG was observed. Fc treatment resulted in inhibition in glucose uptake in both the cell lines, figure 8a. Similar results were obtained when a glucose oxidation assay was performed using culture supernatants from cells treated with increasing concentrations of Fc, figure 8b. Expectedly, Fc treatment also resulted in reduced lactate levels, figure 8c. Hence, Fc-induced cell death appeared to be related to inhibition of glucose uptake. Figure 8. Fc alters glucose uptake and impairs enzymatic activity of HKI in NSCLC cells. A549 or H460 cells were treated with 1mFc for 4H and uptake of the fluorescent glucose derivative 2-NBDG was examined thereafter by fluorescence microscopy. Since an increase in soluble tubulin and both p53 induction and NF-kappa B downregulation, reported earlier 35, have been linked with glucose metabolism, the expression of genes involved in the process was examined. An increase was observed in the mRNA levels of glutaminase 2 and proline oxidase 48H after Fc treatment, while GLUT4 and hexokinase 2 showed considerably reduced expression. p53 inducible proapoptotic genes, TIGR and SCO2 were also induced to an appreciable extent following Fc treatment, figures 8d and S4. Hexokinase 2, HKI, a key glycolytic enzyme, plays a critical role in glucose retention and metabolism and is highly advantageous for cancer cell survival and proliferation. Therefore, we examined whether Fc impairs the enzymatic function of HKI in cancer cells. An enzymatic assay for HKI activity was performed after treating NSCLC cells with Fc for 24H, figure 8f. The results showed reduced HKI activity in Fc-treated cells, suggesting the inhibition of HKI activity to be a causative factor for reduced glucose uptake in Fc-treated cells, eventually leading to the activation of apoptotic signals. In silico models indicated that this action may be due to the ability of Fc to mimic glucose or glucose 6-phosphate, G6P, by stably binding to its pocket in HKI, Fig. S5 and Table S1. The result was further validated by in vitro hexokinase assays, figure 8G and H. Various benzimidazole compounds have been shown to be highly effective as inhibitors, up to 50% reduction of activity, of the helminth-specific enzyme fumarate reductase in vitro. While fumarate reductase converts fumarate to succinate in microbes and lower organisms, succinate dehydrogenase catalyzes the oxidation of succinate to fumarate in mammalian mitochondria. The two enzymes, therefore, act on the reverse directions of the same enzymatic interconversion. Succinate dehydrogenase, SDH, is a mitochondrial tricarboxylic acid, TCA, cycle enzyme and a known tumor suppressor gene 42. Succinate, a TCA cycle metabolite, is accumulated due to SDH downregulation and provides cancer cells with a growth advantage leading to tumor progression 42. Spectrophotometric assay as well as histochemical staining 43 showed enhanced SDH activity in cells following Fz treatment, figure 80I and 2. Combination effect of Fz with other drugs. Finally, we evaluated the effect of Fc in combination with the microtubule targeting drug Taxol, glycolytic inhibitor 2-deoxyglucose, 2-DG, and dichloroacetate, DCA, a pyruvate dehydrogenase kinase inhibitor which acts by shifting the metabolism towards glucose oxidation over glycolysis. In order to determine whether Fc could show an additive or synergistic effect with these drugs, cell proliferation assays were performed and the combination effects were analyzed using the combination index, CI, method 44. The FACI plot for the combination showed that the CI values were 1 over the entire range for combination with DCA. CI value was 0.04 at the 50% effective dose, suggesting a strong synergism by Fc and DCA. Fig. S2A, similarly, CI values for the Fc2-DG combination were also less than 1 for the higher doses with a CI of 0.21 at the FA value 0.5. Fig. S2B. CI1 was also observed at most doses of Fc-Taxol combination, CI 0.52 at 50% inhibition. Fig. S2C. Since CI1 represents synergism as per the CI method, we conclude that Fc shows synergistic effect with DCA, 2DG, and over a range of doses with Taxol. Fc effectively inhibits colony formation of human NSCLC cells in culture. The effect of Fc on cancer cells in vitro was examined by the colony-forming ability of A549 and H460 cells in culture. Treatment of these cells with 1-Fc for 48H resulted in significant reduction in number of colonies as compared to control untreated cells, figure 9a. Further, anchorage-independent growth of control and Fc-treated H460 cells was evaluated by soft agar assay. Results of the soft agar assay correlated with colony formation data, Fig 9bc, suggesting that Fc is a potential antineoplastic agent that kills cancer cells in vitro. Figure 9. Fc treatment results in reduced tumorigenicity in vitro and in vivo. A colony formation assay for H460 and A549 cells following treatment with 1-Fc for 48H, B. Soft agar assay following Fc treatment. Cell colonies were counted after staining. In vivo tumor suppression by Fc treatment. The therapeutic activity of Fc in vivo was examined by giving oral doses to nude mice bearing A549 xenografts. Female athymic nu/new mice were xenographed with A549 cells, and mice bearing tumors, 2 to 3 millimeters, were fed with Fc, 1 milligram per mouse, orally every second day for 12 days, figure 9d. At the end of 12 days, tumors were excised, measured, and weighed. Fc administration led to a marked reduction in tumor size and weight, Fig 9e-F. Further, the tumor vascularity was quantified in control and Fc-treated mice by spectrophotometrically measuring hemoglobin content, Fi 90 nm. Fc administration led to a reduction in hemoglobin content in tumors signifying reduced tumor vascularity, figure 9g. Tunnel staining of tumor sections showed a significant number of apoptotic cells, figure 9H I and 4. These results suggest that Fc inhibits tumor cell growth in vivo by inducing apoptosis of tumor cells. When tumor sections were further examined for p53 protein expression, a number of p53 positive tumor cells were visible in Fc-treated mice, suggesting p53-induced cell death, Fig 9H2 and V. Moreover, Fc-treated A549 tumors showed very few CD31 positive endothelial cells in xenografts, figure 9H3 and 6. These data are in good agreement with our in vitro analysis of Fc-mediated cell death. Discussion. Microtubules are major components of the cytoskeleton and play important roles in a variety of cellular processes like intracellular trafficking, maintenance of cell shape and structure, polarity, cell signaling, and mitosis. Microtubule targeting agents, MTAs, are being used clinically for the treatment of multiple tumor types, but their effectiveness is often largely affected by drug resistance mechanisms. Although the principal mechanism of MTA cytotoxicity relates to their interactions with tubulin and disruption of microtubule function, differences in their tubulin binding characteristics alter their modes of action with a significant impact on the efficacy or toxicity profile of each agent. Newer agents with improved efficacy, tolerability, and the ability to even partially overcome resistance could be of great significance. The present data reveals Fc as a moderate microtubule targeting agent causing mitotic arrest followed by cancer cell death. Despite being a relatively mild microtubule targeting agent, Fc possesses a unique ability to induce p53 to a considerably high level. In our earlier work we have shown the activity of Fc as a proteasomal interfering agent 35. The present study revealed an early elevation and stabilization of cyclin B1 levels in response to Fc, indicating a progression of cells towards cell death rather than mitotic exit and polyploidy. Interestingly, the FDA-approved proteasomal inhibitor bortezomib has also been reported to induce mitotic cell death in lymphoma cells 45. Therefore, proteasomal inhibition associated with mitosis-selective therapeutic approach adds to the significance of Fc as a potential anti-cancer agent. Benzimidazole compounds are known to interfere with energy metabolism of the host, particularly the carbohydrate metabolism. They block the glucose uptake and ATP formation, which ultimately leads to the death of the parasite. Disparity in energy metabolism between normal and cancer cells has been well known for a long time, and in many malignant cell lines glucose consumption is several folds higher than in normal cells. Deprivation of glucose uptake, therefore, has been exploited as a therapeutic approach. 46. Fc could effectively inhibit glucose uptake in NSCLC cells, suggesting that Fc-induced cancer cell death is, in part, facilitated by blocking glucose uptake of cancer cells. Enhancement of oxidative phosphorylation and or inhibition of glycolysis by certain agents, example dichloroacetic acid, 3-bromopyruvate, have been reported to result in tumor cell death. 47-48. Since unlike normal cells, cancer cells mostly thrive on increased glycolysis for generation of ATP, impairment of this pathway could lead to fatal consequences specifically for these cells. Also, it has been postulated that free tubulin regulates mitochondrial function in cancer cells but not in non-transformed primary cells, via alteration of mitochondrial membrane potential. 49. Therefore, microtubule targeting agents having the ability to affect glucose metabolic pathways can be immensely beneficial as anti-cancer agents. Fc exposure reduced the expression of GLUT4 transporter as well as hexokinase, HK2, which may be linked to p53 activation and alteration of microtubule dynamics. 50-51. Hexokinase 2 is a key glycolytic enzyme, which, besides acting to promote glycolysis in cooperation with the GLUT transporters, also acts to suppress mitochondria-induced apoptosis. 52. Targeting this crucial enzyme is therefore being investigated as a possible strategy to effectively curb cancer cell growth. Similarly, proline oxidase 53, 54, SCO2 55, 56, TIGR 42, 57, 58 and glutaminase 2, GLS2, 59 are all p53 target genes involved in the regulation of cellular metabolism, which were found to be induced following Fc treatment. Therefore, tubulin depolymerization and p53 induction caused by Fc may further be leading to modulation of glucose uptake as well as glycolytic pathway. Altogether, our findings show microtubule disruption, p53 stabilization and interference with glucose metabolism as collective underlying mechanisms of Fc-induced preferential elimination of cancer cells both in vitro and in vivo. Materials: Fenbendazole, Fc, 3-4,5-dimethylthiazole 2-whale-2,5-diphenyltetrazolium bromide, emtid, colchicine, Taxol, JC1, 5,5-6, 6-tetrachloro-1,1,3, 3-tetrabethylbenzimidazole carbocyanine iodide, Hooks 33342, propydium iodide.
손 들어오세요.